The trial view window of the data viewer interface provides data viewers many data visualization options and has three main views: the temporal graph view, the spatial overlay view, and the animation playback view. In this video, we'll discuss these three main views. Please see other videos in this series for more data visualization possibilities, including dynamic and difference fixation maps and aggregate data visualizations, which allow you to view the data from all trials and participants in a group or condition at one time. The three buttons at the top left corner of the trial view window allow you to select its current view. By default, each of these three views will show the contents of the trial that is currently selected in the inspector. If you're in aggregate mode, then the spatial overlay and animation playback views will show the data from all of the trials of the group, as we'll discuss in another video. The first button on the left selects the temporal graph view. The temporal graph view is organized such that the x-axis is time and the y-axis represents the XY gaze location in screen pixels, pupil size in arbitrary units, acceleration in degrees per second squared, and velocity data in degrees per second. You can click the data visibility buttons at the top of the interface to toggle the visibility of fixations, saccades, blinks, samples, messages, button events, input sync signals if they were sent to the host PC, and interest areas. As you toggle each button on, its corresponding iData will be displayed below in the trial view window, and event data will be displayed in the inspector as well. You can click on a particular event in the inspector to select it and see its specific data values in the bottom panel. When sample data is visible, you can use the buttons at the bottom of the trial view window to toggle the visibility of different aspects of the sample data, including velocity, acceleration, X and Y position traces, and pupil size. If your experiment had moving targets and was set up so as to send target position messages to the EDF file upon position changes of the target, then you can see the position of the target over time by toggling the target position visibility buttons. I've got some data from a smooth pursuit study opened here that was programmed in Experiment Builder to send target position messages to the EDF file. So we can see how the target position changed over time and how the eye position compares to that target position. The same position data that is visible here can be exported via a sample report, which we'll discuss in another video. Data Viewer will only display data from the currently selected interest period in the temporal graph view, and the boundaries of the interest period are marked by two pink vertical lines. By default, sample traces outside the interest period will be grayed out. The temporal graph view will be zoomed in to fit the selected interest period by default, but you can use the icons with the magnifying glasses on them to zoom in to a selection or zoom back out. And if the zoom level doesn't allow all the data to fit in the view, then there'll be a slider that allows you to scroll backwards and forwards in time. The second view of the trial view window is the spatial overlay view, which can be selected by the second button from the left at the top left corner of the trial view window. The spatial overlay view displays events and samples spatially, allowing you to visualize eye movements superimposed on the stimulus that was presented during the currently specified interest period. We'll discuss interest periods in another video in this series. The same visibility buttons that are used in the temporal graph view also apply to data in the spatial overlay view. Fixations are represented by circles. The center of the circle is the XY coordinate of the fixation, and the diameter of the circle will increase as duration increases. So, the size of the fixation represents its duration. When saccades are visible, each one will be drawn as an arrow from the start position of the saccade to the end position of the saccade. The other buttons will toggle the visibilities of blinks, the sample eye position trace, the location of the eye when messages, button presses, and input sync signals were received by the host PC, and the visibility of interest areas. You'll only see data from your currently selected interest period in the spatial overlay view, and the background of the spatial overlay view will consist of the stimulus that was presented to the participant during the currently selected interest period, provided that this stimulus information was sent to the EDF file by the program that ran the experimental session. If you used Experiment Builder to present the experiment, then this stimulus information is sent automatically. So once you set your interest period, you should see the stimulus of that period. You can also draw interest areas when in the spatial overlay view using the interest area shape tools at the top of the trial view window. We'll discuss interest periods and interest areas in more details in another video in this series. The third view of the trial view window is the animation playback view, which can be selected using the third button from the left at the top left corner of the trial view window. The animation playback view allows you to see how the eye position changed over the course of your trials, and is especially useful for projects involving dynamic stimuli, like experiments involving videos. You can control the playback speed, and you can use the playback controls at the bottom of the trial view to move to different time points in the trial. You can also create and edit dynamic interest areas in the animation playback view. Again, this will be covered in more detail in a later video. Each of DataViewer's possible views has a Save Trial View as Image button, which allows you to save an image file of what is shown in the trial view window. In the animation playback view, you can use the Save Trial to Video File button to save a video of the trial's playback. In the spatial overlay view and the animation playback view, you can also enable the aggregate mode to view more than one trial's data at a time. This allows you to view all the trials for a certain condition or of a given participant at one time. You can see other videos in this series for more information on grouping trials by variables and on aggregate mode.